medical virology. He's esteemed American Society of Clinical Oncology, a peer reviewer for multiple international journals, and was also awarded the Presidential Poster Award at annual meeting of the American College of Gastroenterology 2022. A warm welcome to Dr. Ibad, and we're very grateful that you agreed to joining us and teaching us the essentials of meta-analysis. Thank you so much. Over to you.
Hi, could someone let me know if I'm clearly audible?
Yes, you are.
Okay, yeah. Hi, everyone. Thank you so much for the kind introduction. So let me just share my screen. We'll go from there. Okay, can you guys see my screen?
Yes, it's visible.
Okay, thank you. Okay. So before I start, I'd just like to, get an idea of the kind of audience that's here. So I would be really grateful if everyone would just, let me know which year of med school you're in the chat very quickly, just so I can get a quick idea of what grade everyone's in. So which year are you in currently? Yeah, fourth year, third year, second year. Okay, that's really good. There's a lot of people from second year, third year, fourth year. I also started research in second year. Back then, there wasn't this trend of actually doing research. So you guys are already really ahead of the curve here. Is this everyone's first? Oh, we have some MDs here too. So is this everyone's first experience with learning a meta-analysis or learning manuscript writing in general? Have you heard of what a manuscript is in the past? Okay. 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 So before we jump onto the slides, I'll just quickly tell you what a manuscript actually is. So, in the previous lectures, you've probably covered how to come up with a topic, how to do screening, how to do extraction, run the analysis, check the risk of bias, right? We've done all these steps, right? So the manuscript is the final step, probably the most important one at that. We'll just go through learning objectives first. So we're going to look at how to write a good manuscript for a systematic review and a meta-analysis. What are the components of a manuscript, and some of the tips that I've sort of learned the hard way over the years. So I'm just going to give you some of these tips. So a manuscript is basically the written report of your research, right? So you've done all these steps, right? You've climbed all these stairs, gotten to the door. The manuscript is where you actually tell the journal, their editors, their reviewers, and the readers who are going to read your publication what you actually did, right? So all the steps that you've covered in the previous sessions, they're all going to get summarized in your manuscript, right? You're basically going to tell them what you did, and it's the very last step. So that's where it's the most important one, other than arguably topic selection, because if you do a bad job with running, if you do a bad job with your risk of bias, or if you do a bad job with your data extraction, that can be covered up in the manuscript, right? If you write it really well, you can cover up your flaws, your paper's flaws. But if your manuscript writing isn't good, then no matter how much hard work you've done in the past, it won't matter, because the thing the journal will see is the manuscript, right? So a manuscript is a written report. I also have an example here. So this is one of my publications from last year. It was a mentorship project. So this was our topic, and this is what a manuscript looks like, right? It's just a Word document. I'm just gonna zoom in a little. So this is just a Word document. It's like 6,000 words. There's different sections where we talk about what our project was, what were the different steps, what databases we used, and everything, right? So this is a manuscript. It's like a very long essay about your paper, right? And then at the end, there's tables, there's also figures. So we're just going to cover all of that. So a manuscript answers 4 main questions: why you did this research, what did you actually do, and then what did you find, and what was the meaning behind what you found out, right? So these are the 4 questions, and they basically correspond to the 4 sections of a manuscript, right? So before we discuss that, so a manuscript is like a story, right? So I'll just ask you guys, trying, it's kind of a dry lecture. It's not as fun as teaching analysis or risk of bias. So I'll try to keep it interactive. So I'll just ask you, you've probably seen movies or read books. What do you think makes a good story? What are the characteristics of a good story or a good movie or a good novel? A good opening scene, the plot, the prose, the language, right? The visuals. Yeah. So a manuscript in essence is a story, right? You're telling them the story of your paper. So I identify, these are the 4 things you want every story to have. It should have a message, right? It should have a purpose. Then it should have a logical flow, the plot basically, right? It should have a beginning, a middle, and end. It should follow in an order. It can't start with the middle and then go to the beginning, right? It has to have a order. It should be relatable to the reader, and it should be understandable to them, right? It shouldn't be confusing to them. So in the similar way, what makes a good manuscript? It has to have a clear and exciting message. It's not going to be a moral message, right? You aren't going to say sharing is caring or something. It's going to have a clear and exciting scientific message. So, the purpose for most people to do a research is the purpose for most people to do research is, for most people it's to add a publication to their CV. But your paper needs to have another purpose to it. Needs to have an actual purpose, like it should answer a research question that's actually relevant to the field, right? Otherwise it's not going to get published just because you want it to get published. Then it should have a logical flow. It should be written in clear language, right? Even if your English is really good, a lot of difficult words, don't use all of them. You want the other guy to understand what you're saying without opening a dictionary, right? And it should be relatable in the sense that it should be connected to the previous papers on that topic, right? If I'm doing a research on blood cancer, I need to talk about what's already been done on blood cancer, right? I need to connect to that. So that's what relatable would mean in this context. So before we start with the manuscript, these are the things we should already have. Hopefully all of them have been discussed before. If something hasn't been discussed, yeah, let me know, we can discuss it. So you should have a search strategy. You should have a Prisma flow chart. You should have a baseline sheet. You should have your forest plots, and you should have your risk of bias or your traffic light plots, right? So the components of a manuscript, obviously the first thing is so we can look at an example that'll be a little easier. Yeah. So the first thing at the very top is obviously the title, right? The title and everyone's names and everything. After the title, the first section is the abstract, right? The abstract is basically a summary. So this section is a summary of all the sections that are going to come after it, right? So these are the subheadings in the abstract. We're going to look at them later. But this is basically a summary. It's like 200 to 50 words, everything summarized, right? From here all the way till here. This is the abstract. After the abstract, the first thing is the introduction, right? You introduce your topic. Why are you actually doing this research? That's the introduction. After the introduction, so if you remember the 4 questions I talked about, the first was why are you doing this research? Then what did you actually do? That's the method section. What did you actually do, right? Then after the method section, what did you find out, which is the results, right? And then after that, you explain what you found out. So you explain your results, which is the discussion, right? And then the discussion ends with a conclusion, which is basically a part of the discussion, right? So this is the conclusion here. So these are the main sections. Right? So the way they are written in a manuscript is the first the title, the abstract, the intro, method, result, discussion, and conclusion, right? But when you actually write them down, you can't follow this order. For example, the abstract is a summary. It's the first section, but you can't start by writing a summary, right? You need to write down the entire thing first before you actually understand what you are going to summarize. So the way they are actually written down is this way. First, you write the methods, then you write the results, then you write the discussion, and then you go back to the intro and the abstract, right? And then usually the best time to come up with a title is the very end when everything's clear to you. So you are going to write them down in this order: method, result, discussion, and so on. But in the actual manuscript, they're going to be written like this, right? Hopefully that makes sense. So the first section is the method section. So there's a lot written here, but method section is simply a recipe for your research, right? So you're going to tell the reader what you did, and the goal is that it should be in enough detail that anyone should be able to replicate it, right? So the basic principle of scientific research is that you report your findings in such a way that anyone can recheck your work to see that, yeah, this was done correctly, right? So even if someone in South America reads it in 2045, they should still have all the information to be able to recheck your work, right? So it's a recipe. There are subheadings in the method section, right? So we are discussing the method section first because that's how you would write it down, right? But in the actual manuscript, the method section comes in the middle, right? So we're going to go through the full manuscript at the end as well, just to, make it all make sense. So this is basically a recipe for your research. So just exercise, I've written down 2 recipes. And I just want you to tell me which one's the better one, the one on the left or the one on the right. It's like a mutton recipe for chops, I guess. Yeah, so people are saying the one on the left, right? It's obvious. The one on the right says mix everything, cook it till water dries, right? The right one tells you, 2 tablespoons of this, 1.5 tablespoons of this, 1 teaspoon of this, right? So recipe needs to be detailed. I'm not a really good cook. I can't copy the one on the right? If someone knows how to do it, maybe they would be able to do it. But for people who can't cook, the one on the right is useless. I can copy the one on the left, right? Because it's very specific. Leave for 2 hours, take 2 teaspoons, right? So in the same way, we want our method section to be really specific on everything that we have done, right? Because the method section is also a recipe. So first thing is the Prisma guidelines. Have you heard of that before? Hopefully you've heard of Prisma guidelines before in any of the previous webinars. Okay, that's really good. So Prisma guidelines or Prisma checklist is basically checklist. Like you have to follow these guidelines to make your systematic review, right? So for the method section, this is relevant because your method section will always start with this sentence. So this systematic review and meta-analysis was done in accordance with the Prisma guidelines, right? So this is how you actually start your method section. You just say that we have followed the Prisma guidelines. Now I added this question, but then I changed the slide. So I was asking you what Prisma would mean, but then I wrote it down. Prisma basically means preferred reporting items for systematic reviews and meta-analysis, right? That's the full form. So you're going to start your method section with this sentence. That's going to be the first sentence. Then the first subheading. So the way you think about the method section is think about your research. What was the first thing we learned? What was the second thing? So we started with the literature search, right? The first thing we started with was the literature search. That was the first step. So we're going to go from that first step all the way to analysis, which was the last step. And we're going to talk about what we actually did in each step. So the first subheading is the search strategy. When you talk about the search strategy, you need to mention, what was the dates of your search? What was the exact search that you used? What were the mesh terms and keywords that you used? What databases were used, and any additional details. So it'll be more clear with an example. So, but before we go to the example, so these are the 4 things, and then additional details. So these are some of the additional details. These are basically just filler sentences that people add to their method section to look cool. But basically, these are the 3 things you can do. Hand searching, which means, if you have an article that's relevant. I'm doing a research on blood cancer, and I find a paper that has been published that is, a paper that I want to include in my meta-analysis. Hand searching means, I've included that paper, but I can also search the entire reference list, all the citations at the end of the paper, right? The bibliography. Every paper has a bibliography at the end. So I can manually search that to see if there's any other relevant articles. That's what hand searching is. The other 2 things aren't that important. So this is an example of how to write down your search strategy, right? So an electronic search was done on, so first thing I'm doing is I'm mentioning the databases that were used. The search was done on PubMed, it was done on Embase, Cochrane, clinicaltrials.gov, right? So I've mentioned the databases. What was the duration of the search? from inception up to August 2022. So inception basically means like the very beginning of PubMed, right? You don't really, I don't say I'm only going to look at articles after 2000. Any article that was ever published on PubMed, all the way up to this date, which is, basically when I was working. So when you sit down and I run a search on PubMed right now, I will include articles from inception to July 2025, right? So this is the date of my search. The next thing was what were the mesh terms? Hopefully you've heard of that as well. What were the mesh terms that were used? So you can just mention the mesh terms: total body radiation and acute lymphoblastic leukemia, right? So this is one of my older projects. I mentioned that as an example, right? And then you can also mention the entire search string. So you can just add a table, just copy paste the entire search that you used, just copy paste it into a table, and you can mention your full search string like this. It doesn't make sense to include it in the actual paragraph because the search strategy is like really long. It's going to break up the paragraph, but you can add it as a table, right? And then this was some extra stuff that this is like filler, right? Just to make your method section look cool. So I went through this conference, American Society of Hematology. It's like the biggest hematology conference in the world. So I went through their annual meetings to see if there were any relevant articles, right? So this is how you write down a literature search. The main things you need to focus on is name the databases, the duration of the search, and the mesh terms that were used, right? So after you did the literature search, the next thing was study selection, right? Screening, which we did on, I'm assuming you guys did it on Ryan, right? But there's a couple of different software that can be used. So for screening, now when I need to actually write down the screening in my method section, I'm going to talk about which software was used. So, EndNote, Mendeley, Zotero, Ryan, whatever was used, right? Then you need to mention the criteria that was used, right? So what was the inclusion criteria? What was the exclusion criteria, right? So I included patients with blood cancer who were given, this drug, who were given a tyrosine kinase inhibitor. That's a kind of chemotherapy, don't get confused. So that's your inclusion criteria, which is the same criteria that you used for screening, right? And then another thing is, I don't know if this has been discussed in screening or not, but Prisma recommends that 2 people do screening at the same time, right? So you always need to write down a sentence like this: 2 authors independently screened the titles and abstracts, right? And checked the full text, and disagreements were settled by a 3rd author. This is just based on the Prisma recommendations. So if you look at an example of this, all articles were imported into EndNote or Ryan or whatever you used, right? Then 2 authors, these are their initials. For example, my name is Muhammad Ibadur Rahman. So Muhammad Rahman, these are my initials, right? So 2 authors independently screened the titles and abstracts, right? This is basically primary screening. You're talking about that you did primary screening. So 2 authors independently screened the titles and abstracts and removed articles that were not relevant. Then full texts were checked, and then conflicts or disagreements were settled by a 3rd author, right? So this is how you write down screening. Here we are talking about the actual inclusion criteria, right? So we included RCTs. So if you've heard of the word PICO's framework or PICO's table, this is basically the PICO's framework for this project. We included RCTs, so that's the study design we are including. Patients who had leukemia, right? Blood cancer, simply. So this is the population that we are including. And we are comparing radiation or radiotherapy simply. I know this is a really complex example, but I'm trying to simplify it for people who are in 2nd or 3rd year. So comparing radiotherapy with chemotherapy, right? So RCTs, including patients with leukemia, comparing radiation with chemotherapy. These are population, intervention, comparison, and the study design, right? And then the exclusion criteria is simply the inverse of the inclusion criteria. So it's just going to be study designs other than RCTs, such as animal studies were excluded, and there were no language restrictions. You can choose to restrict yourself only to English. It's up to you, right? So this is how you write down the screening, which was the next step. After screening, the thing you did after screening was data extraction, right? So in data extraction, just mention the variables that you extracted, right? You made a Google spreadsheet probably. So just mention what variables were extracted, right? Talk about your outcomes. What were the outcomes that were extracted, right? And then in the next thing after that was risk of bias assessment. So just mention what tool you used, mention its domain, and then again mention who were the authors, their initials, who actually did this stuff. So this will be more clear with the examples. So data extraction was done by two researchers, again their initials, disagreement was settled by a third author, right? So this is just a generic sentence you can add to every section of the method section. Now after that, it's simply just a list of all the variables that you extracted. So information obtained included first author, year, country, study design, and so on, right? And then I'm mentioning all the outcomes that were extracted. They're like complex, so we don't need to understand them, but this is just, I'm just listing down the outcomes that were extracted. And for risk of bias, again, two authors assess the risk of bias using this tool. Hopefully you have heard of this, ROB2, right? This is the full name. So you write down the full name, the Cochrane Risk of Bias Tool for Randomized Trials, ROB 2.0, right? And then you can mention the domains of this tool. Have you worked with ROB2? Has that been covered in a previous lecture? Okay, it has not, which is surprising. Okay, then I don't know how to make sense of this. But yes, it's basically a tool to check the quality of a study, and these are the domains. So if you know, if you use that tool, you should mention the domains. And then the basic thing is, disagreements were discussed and resolved with the third author. So the last thing, I'll go through this quickly because I think a full example of a method section will make more sense. But for data analysis, which I think has been covered using RevMan most likely. So just mention what model, what method was used, mention the software that was used. So instead of this, we're just going to look at a full example. So this is the paper. Now we're going to look at what the full method section looks like, right? And again, we start from the very beginning of the research and go all the way to the end. So method section starts here. This sentence you always add, right? Our meta-analysis was conducted according to the Prisma statement, and you can say we followed the Cochrane handbook. That's also another guideline to do a systematic review. What I am teaching you is in accordance with the Prisma statement and Cochrane handbook. So basically, if you follow my lecture when you work on your projects, this statement is going to be true, right? You will have followed the Prisma as well as the Cochrane handbook, right? So just mention you followed these two things. Then you'll mention that you registered your paper on Prospero. We will discuss what Prospero is at the very end, right? After that, you start from the very beginning. The first thing you did was literature search. So we performed a literature search on Cochrane. Medline is basically PubMed. This is another name for PubMed. So Cochrane, PubMed, Embase, and clinicaltrials.gov, these were the databases that were used. This is the duration of the search. And these were the mesh terms. So this research was on dialysis and angioplasty, right? So these were the mesh terms that were used. So here I'm talking about the literature search. The next thing, you can make it a separate heading. This is basically the inclusion criteria and the exclusion criteria. So study design needs to be RCT, population has to be dialysis patients, and I'm comparing two different interventions. They don't really matter, but just as an example, this is how you write this down. And then what needs to be excluded is cohort, case control, cross-sectional case series, review articles, right? So just the opposite of the inclusion criteria is excluded. The next thing is, so here I'm talking about the screening. So again, we imported our articles onto Ryan for screening. Two authors did it independently, and the third author was there for disagreements. So screening is this section here. And then usually I talk about data extraction in the same subheading, but it's up to you, right? There's no hard and fast rules. For data extraction, just make a list of all the outcomes and all the things that were extracted, right? So this, depending on your research, this is just going to be a list of your variables. Risk of bias, which maybe wasn't covered, but so the way to talk about risk of bias is you name the tool and you name all of its domains, right? And then finally, data analysis. So for data analysis, you need to mention what software was used. So if you use RevMan, this is the full name for RevMan, Review Manager, RevMan 5.4, Cochrane Collaboration, Denmark, right? This was the software that was used. And then you talk about what you actually extracted, right? So hopefully you've covered this, but we extract a risk ratio for dichotomous outcomes and mean difference for continuous outcomes usually. So here in this example, the risk ratio along with the confidence interval were extracted for dichotomous or categorical data. This is the model that we used for dichotomous data. So this is the model you write down, right? And then I-square statistic was calculated and forest plots were made, right? So this brings you to the end of the method section. Is this clear? Any questions? If it's clear, you can let me know. If anything isn't clear, you can let me know before we move on to the next section. Okay, it's clear to 2 out of 70 people. Okay, so after method section, yeah, so this is just an exercise, but we'll skip it. So the next section you write down is the result section, right? You actually talk about what you found out, right? So in the result section, you talk about what you found, probably the most important section. This section also has subheadings. So the way the method section works is you start from the very beginning, from your literature search, go all the way to analysis and tell what you actually did. In the result section, you do the same thing. Start from the very beginning, go all the way to the end and talk about what you found out, right? For example, when I make a literature search, right? The terms I used, which database I searched, everything, that's what I did. That belongs to the method section. How many articles I actually got, that belongs to the result section, right? So every step is split into the methods and the result section. Um, for running an actual analysis, what model, what software you did it on RevMan, what you used, that belongs to the method section. What the actual results were, what was the actual forest plot, belongs to the result section, right? So hopefully this makes sense. Every step that you have covered in previous lectures, it gets split into two. We're going to get to the discussion section. Discussion section is basically an explanation, so we're going to cover that. But again, now I'm starting the result section, so I'm going to start from the very beginning again. I'm starting from the very first thing. So this is a golden rule for any type of research that you do, meta-analysis, narrative review, if you do clinical trials in the future. Every result section, the focus is always on the tables and the figures, right? Picture is worth 1,000 words, because it's easier for people, especially people who are used to reading researches. They just, when I read a paper, I just look at the tables and figures, right? So the focus is always on the tables and figures. So any type of research you do, how will you write a result section? Even if you're doing a cross-sectional study or a cohort study, first you make all your tables and figures, and then you basically just write a caption to explain them briefly, right? The way when you post something on Instagram, the focus is the picture, right? And then you just add a caption to go along with it. In the same way, the full detail is always in the tables or the figures, and the caption is just a quick brief explanation, right? So just 2-3 lines to describe every table and figure. So again, when I start with the meta-analysis, the first thing I do is I do a literature search and I do screening, right, on Ryan. We have covered this, hopefully. For have you looked at a Prisma flowchart before in a previous session? Okay, okay, that's really good. How to, so this is a Prisma flowchart. Hopefully you've covered how to fill it in, right? So the very first section in a result section, the very first subheading of the result section is called search results. This is basically your literature search as well as your screening. And we have already covered that this is summarized in a Prisma flowchart. So the figure associated with this paragraph, right? Every paragraph will have a figure. The figure here is the Prisma flowchart. And then you just write down in the actual text, you just briefly explain it. So if after you've made the Prisma flowchart, this is what an explanation could look like. So I've written down 2 of them, and you can tell me which one is better, right? The first one says literature search retrieved 1712 studies after exclusion of duplicates, reviews, and articles. A total of 11 were included. The literature search process is summarized in figure 1. So this is the first example, right? Figure 1 is basically the Prisma flowchart. So this is one example. The other example says a strategy retrieved this many articles after removal of duplicates, titles, and abstracts were searched, then full text were searched, and finally 15 studies were included. So which one do you like more, the first one or the second one? Which one do you think is correct? Okay, so this is basically a trick question. They are both correct. The people who say the first one, they probably like it because it's really concise, right? The people who are saying the second one like it because it's detailed, it tells everything. They are both correct. I would personally go with the first one, because if someone wants to read the full detail, what will they look at? They will look at the figure. The figure has all the detail. There's no point in saying all of that in the words. But the second one is not wrong. People do the second one as well. So both of them work, right? But hopefully you get the idea. So this is, so after you made the Prisma flowchart, just a brief explanation of the flowchart, right? You started with this many articles, you ended with this many articles, and you're done with your study selection. So you basically covered literature search as well as screening for your result section. Okay, so the next section is the study characteristics section. For some reason, my slide isn't working for this section. So we're just gonna do it directly through the example first. So after screening, the next thing we did was data extraction, right? So now we need to write down the results for the data extraction. So how do you write that down? The main, what do you think would the main table or the figure for data extraction would be? If you've covered this, because I said every paragraph in the result section has a table or a figure. So what do you think the figure or table for the data extraction would be? Any guesses? Okay, no guesses. So basically when you do data extraction, you make a baseline table, right? It looks something like this one, right? Or if you want me to just pull up a data extraction sheet, I'll do that as well. So I make data extraction, yeah, the spreadsheet. So this is another mentorship project. It's not complete, but this is what a data extraction sheet should look like, right? There's a baseline sheet. This is your baseline sheet, right? How many, what was the average age, what was the sample size, what was the country, right? Maybe this is a more complete example. Yeah. So this is your baseline sheet. You will simply copy it and make a word table out of it. So for this example, you can see this is a word table in Word. All of that is just copy pasted from the baseline sheet. So this is your table for this paragraph. In a manuscript, you put the tables at the end, and you know the name is called table 1 characteristics of included studies, right? So this is the table. And then I just need to briefly describe some of the characteristics. So I will say, okay, I included 15 studies. Total number of patients was this. And then you can talk about, you know, how many studies were done in which countries, how many people were in the intervention group, and how many were in the control group. What was the average age like? What was the gender distribution, right? Just pick some of the characteristics and briefly explain them. You don't need to go into too much detail, because if someone wants that, they can just read the table. And then at the end, you say study characteristics are presented in table 1. So people know where to go, right? So this is what the data extraction covered in the result section looks like. After data extraction, so was risk of bias not covered? I'm not sure how to discuss this now. Was there a webinar or something on risk of bias assessment? Okay. Okay, it hasn't been taught yet. It is optional, so you know, maybe that was the point. But if you do have risk of bias in your meta-analysis, you just briefly explain it like this. It's kind of hard to explain this now without that context. So we'll go to the next section. So after risk of bias, the last thing in your result section is going to be the analysis, right? The actual meta-analysis. Before that, I'm going to ask you I have, because I'm hoping forest plots have been covered. So this is a forest plot. This forest plot is looking at relapse, right? I want you to interpret this. Relapse. What does relapse mean? You know, when someone's cancer comes back. I mainly do oncology research, so that's why all these examples are oncology. So when someone's cancer comes back, that's what relapse is, right? And I am comparing radiation. This is the radiation group, TBI, total body radiation, and chemotherapy, CHT, right? Chemotherapy. So these are the two things I'm comparing. The outcome is relapse. I want you to look at this forest plot and tell me, are these results significant? Okay, and why are you saying they are significant? Okay, okay, okay. So Adele is saying it doesn't cross the midline. He is talking about this diamond. You are right. When this diamond touches this line, results are insignificant, right? But obviously, you know, in some cases it's really close. You can't judge. In this case, it's very clear, but sometimes it's really close. So the best way is to look at the p-value, right? P-value is, so this is the p-value, right? Test for overall effect. P is less than 0.00001. What is the cutoff for p-value? What should the p-value be less than for results to be significant? 0.05. It's not 0.01. It has to be less than 0.05, right? This is much less than 0.05. So these results are significant. Now I'm going to ask you which group is better, right? Which group prevents relapse more, radiation or TBI, right? That's what TBI, I wish I wrote it down as radiation to make it easier, but is radiation better or is chemotherapy better? Yeah, so radiation is better because, you know, the first thing is the graph tells you TBI, it favors TBI. The other way is you look at the risk ratio, right? 0.69. When this is less than 1, that means radiation is better. If it was more than 1, that would mean radiation is causing relapse instead of actually preventing it, right? So yeah, you guys know this really well, which is good. So now we need to actually write down, like I have this forest plot, I need to write down results for this forest plot, right? Now I just need to come up with the English. We understand everything, we just need to make a sentence. So how do I make this sentence? The first thing is you will have different outcomes. So first just talk about the most important one. Like in most researches, mortality is the most important thing, right? Because the first thing you want is for the patient to, you know, be alive. So just arrange your forest plots, and then for each forest plot, you're going to write down the number of studies in that forest plot. How can you tell? So, you know, just count them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is like 13, 14 studies, right? Every row is one study here. So just count these and you'll know the number of studies. So number of studies in that outcome. Then you're going to talk about your pooled result. So the way you write this down is first you write down the value of the risk ratio. Then you write down the confidence interval, then the p-value, and finally the I-square value, right? So all 4 values in a row. So for this example, what I would write down is 13 studies reported on relapse as an outcome. TBI was associated with a significantly low risk of relapse compared to chemotherapy. And then basically I just write down this sentence telling people which one is better, and then just open a bracket, write down the risk ratio, write down the confidence interval, write down the p-value and the I-square. Just list all 4 values down. That's it. You have written results for one forest plot. So this is a second forest plot, right? And for some reason, I still say relapse here. I should have said non-relapse mortality. So non-relapse mortality is another oncology outcome. You don't need to understand what this means. But basically, I look at this forest plot. These results are significant. Radiation is better. And I will just write that down. 11 studies reported on non-relapse mortality. Radiation was significantly so low risk of non-relapse mortality. And then just list down all 4 values, right? This is a forest plot that is insignificant because the p-value is more than 0.05, right? So in this case, I'm going to say both groups were comparable, right? So 7 studies reported this outcome. Graft versus host disease is basically a side effect, right? So TBI-based regimens were comparable to chemotherapy. So radiation was comparable to chemotherapy. Why? Because the p-value is more than 0.05. And in the same way, I just list down all 4 values, right? So that's literally it. That's all you need to do for the forest plots. So let's look at an example. For a full result section now. So this is result section. It starts with the search results, which is the literature search as well as the screening. And again, this many articles were obtained. Primary screening, secondary screening. Finally, this many articles were included. Figure 1 summarizes the screening process. Figure 1 is the Prisma flowchart. We have already discussed that. Then the next thing is data extraction, which is written under the heading of static characteristics. 27 studies were included, this many in each country, and total patients was this many. Year of publication ranged from here to here, and then number of people in each group, and table 1 has all the details. So you notice at the end of every paragraph, I'll say, go to this figure, go to this table. That's how you write result section. Risk of bias we haven't covered, so we'll just skip this. And I'm just going to find a convenient example which is easy for everyone to understand. Okay, so all cause mortality, right? So the way you write down the result section is, just make a heading for each outcome. This is first outcome, second outcome, third outcome, fourth outcome, right? All cause mortality simply means mortality, right? How many people died. So in each outcome, I write it in the same way. 11 studies reported data and both groups were equal. And then just open a bracket and write down all 4 values. And after that, the figure here is going to be the forest plot. So we'll just mention that, this is the figure where you're, so people know if they want to see all the details, they can look at the forest plot. Sometimes I can combine outcomes. So for example, I was looking at an outcome called TLR. We don't need to understand that is, but I was looking at TLR. And I looked at this outcome at 3 months. I looked at it at 6 months. So after these patients underwent a procedure, and I looked at the outcome after 3 months, after 6 months, after 12 months. So because all of these outcomes are very related, the only difference is the time period. So I made them all into one paragraph, only one heading. So that's up to you, right? They were similar. All these headings seemed like very small, like, very one sentence and then another heading and so on. So I just combined all of them. So that's how you write down the result section, because after analysis, you've basically completed all the steps. So is the result section clear, or are there any questions? Yeah, I know it's a really dry lecture. There's nothing I can do to make it more entertaining. So the next section after results is discussion, right? If you remember the order, right here, right? In the actual, this is the manuscript order, but we are writing them down like this. So we've written the method section, we've written the result section. Now we are on the discussion, and then we'll go back to write the introduction, right? And the abstract. So in the discussion section, so the discussion section is the most open-ended section. When I started out, no one, told me what you write down in a discussion. It's the most open-ended thing. Basically, it's like 1,000 to 1,200 words, and you just explain your findings. For example, when you look at this paper, after the results is done, you start with the discussion, and then you can see this. It starts here. There are no subheadings, no nothing. It goes on and on and on and on, explaining the results, right? And then at the end, there's a conclusion, but in the middle, there's this large chunk which is just open-ended. And it's, I hate things that are open-ended. I have always preferred having a framework to follow. So that's what I'm going to try to give you. But at the end of the day, you have to remember that discussion is the most open-ended thing. There's like no wrong way of going about it, as well as long as you cover everything. So I have written down 4 things here, but there are no subheadings. In the method section, there were subheadings. In the result section, there were subheadings. The other sections do not have subheadings, right? So in the discussion, you basically explain your results. Why are they like this? So you provide context, compare them with the previous studies, and try to give reasons. So the first thing you do is you summarize your findings, right? A very quick recap of all the results without writing down the values, the p value, the risk ratio, all the values, just a quick recap what the actual results were, right? That is a summary. If you want to look at a summary, this is a summary, right? The first paragraph of the discussion is a summary. In this meta-analysis, we looked at RCTs comparing these two things, right? Total this many RCTs were included, and then you just say that these outcomes were significant and there was no statistically significant difference for these outcomes, right? So you just mentioned these were the significant outcomes, these outcomes were insignificant. No need to write down the p-value and everything because that's already written in the result section. So that's what a summary looks like. After the summary, you basically discuss your findings, right? You will take each outcome, right? It's actually up to you how many outcomes you take, but you take at least 3 or 4 of your outcomes and you explain them. Why are they like this? So that's what I mean by discuss findings. So you start with the most important outcome called the primary outcome, and then you move on to the secondary outcome and so on. So that's the second thing, discuss your findings, right? Before we look at this in more detail, because this is basically the bulk of the discussion. After the summary, the first paragraph's the summary, all this is basically discussing your findings, right? Except for the last two paragraphs, right? So we are going to come back to this, but let's quickly look at what the last two paragraphs are. So the last two, this is discuss your findings. So the last two paragraphs are the limitations. You talk about your limitations, also try to mention you covered them. For example, limitations in methodology, limitations in the review, any populations that were not included. With every type of study, you have to mention your limitations. And then the final thing you write down is the conclusion, right? We'll look at examples of these later. So these are the 4 things in a discussion. Now I'm going to look at specifically discuss you, because this is like the bulk of the discussion, right? So we are going to talk about how we actually discuss a finding. What you do is pick one outcome. For example, let's choose, the example we were looking at. Let's choose a relapse. That's my main outcome. I choose relapse. So I am going to write down one paragraph on this outcome only, right? How do I write down this paragraph? I am going to start right here. But it's a circle, which is confusing, but it starts here. First thing you do is you are going to, first you just quickly recap your results and then compare with any previous meta-analyses that have been published. Compare with any major clinical trials. Then try to identify any reason for similarities or differences. Then you can basically talk about pathopharma, all the basic sciences stuff. And finally, you talk about what the impact of these results is, right? So these are the 5 things. That's a lot. Let's look at an example to make it clear. So let's look at the easy to understand example. Okay, so here, this is one paragraph on this one outcome, right? This entire paragraph is just on one outcome. Now the outcome is TLP. You don't need to understand what TLP is, but it's basically the degree of vessel opening, right? Patency of a vessel. But again, we don't need to understand the actual research that I did because I've already published it. So this is the outcome. I chose the outcome first. Then this entire paragraph, how did I write it down? I wrote down first my own findings. Very quickly in one sentence, just a quick recap. I've already done a recap, but now I just talk about this outcome specifically. What were my findings, right? So in yellow are my findings, right? Then I start to compare with the previous meta-analysis. You can see similar to our findings, a recent meta-analysis by this person showed the same results, right? So I'm going to highlight the comparison part in green. So this is one meta-analysis. Then I found another meta-analysis. I compared my results with that. And basically my results are the same as the previous meta-analysis, right? So first you talk about your outcome. Then you compare with the previous meta-analysis, as many as you can find. And then you try to look for reason for any differences, right? So these are some reasons. They don't really matter because, they're like dependent, they're like specifically about my research. But I talked about, okay, we all included RCTs, large sample size, and so on. This is why the findings were the same. If the findings would have been different, if the previous meta-analysis says relapse is insignificant and I say relapse is significant, try to find out why, right? Did you include any extra studies, right? So this is sort of the reason. Sometimes it's really difficult to find a reason, so you just sort of gloss over this part, you sort of skip this part. But if you do find a reason, you can mention it. After that, you can basically talk about some of the bookish knowledge. Now obviously, again, the discussion section is open-ended, but basically you talk about, let's make this pink, right? So you talk about, okay, how was this drug actually acting and so on, right? Like the patho, the pharma, the pharmacokinetics, pharmacodynamic stuff related to your topic, right? Which is causing these results, which is like how is radiation actually preventing relapse? We said it does, but how, right? It causes DNA damage and etc, right? So that's like the bookish stuff. And then you can talk, it's up to you, but you can talk about how this will impact practice, right? For example, chemotherapy was used a lot, but radiation is cheaper, so it should be used more, something like this, right? So that's the general structure, just to give you it's like a framework. You don't have to follow it every time. Let's see if there's another easier example. So I do have another paper that had a much better example, so I'm gonna pull that up really quickly. So this is another research. Again, we don't need to understand this research, but just as an example. So this research was on nephropathy, right? Renal injury, basically. So this one paragraph is only on nephropathy, contrast-induced nephropathy. CIN is basically damage to the kidneys, very simply, right? So that's what this research was on. So this entire paragraph is only talking about this one outcome. The first sentence is a quick summary of my results, right? Our meta-analysis shows this, right? Then you start with a comparison. Previous studies support this finding. For example, this study had 7 RCTs in it, and they found this. Another meta-analysis by this person had 5 studies, they found this. Another meta-analysis by this person had 14 studies, they found this. All results for the same outcome. Don't talk about another outcome here. The entire paragraph is on one outcome. So all this is the comparison with previous meta-analysis, right? In green, and let's make it like this. And then because all the results were similar, I don't need to give a lot of explanation, right? Because it's expected for all the results to be similar. So this is this explanation, just mention, just sort of like a conclusion that all the meta-analysis have the same findings. And then you can talk about, like, this is like a pathology, incidence, high risk, it acts on, like, it causes this is the mechanism, nitric oxide, and so on, right? So this is sort of the bookish stuff, right? So this is how you write on one paragraph. After this one paragraph, pick another outcome and write on that outcome. For example, here, adverse events. This is my next outcome. So again, I'm going to repeat everything: my summary, my results, comparison with the previous meta-analysis, and then sort of any explanation, any mechanism of action and stuff like that, like dilation and ischemic preconditioning and so on. So you repeat this for 3 or 4 of your outcomes. This is what this circle is, right? Hopefully that makes sense. Again, at the end of the day, this is still the most open-ended thing in the entire manuscript. After that, you come to the limitations and conclusions. So let's look at limitations. So this is a separate paragraph. Our study had several limitations, and these are going to depend on your topic, right? For example, our RCTs were not double blind. There were different confounders, like, age was different and so on. So these obviously depend on your topic. And we had very high heterogeneity and stuff like this, right? And after that, the conclusion is a quick recap of your results again. The same way you started discussion with a quick recap, you end it with a quick recap too. And then you talk about what further studies need to look at. Further studies will look at DCB, they look at cost effectiveness and so on, right? So this is what the conclusion is. But even then, discussion always stays the most open-ended and the trickiest section to write. So we'll look at the introduction. This is easier. again, I have managed to mess this up. But so intro for every paper is like a funnel. It's like a funnel because you start broad and you narrow down, right? So at first you're going to talk very broadly about your topic. Then you're talking about what is known, what is unknown, what is the rationale for your project, and what are the objectives, right? So you need to give a quick overview. The most important thing is, talk about the global burden. It's a very common disease. Why is this be like this? So it's a very common disease. It has very high mortality, morbidity. You basically need to sell your research in the introduction. So you're going to talk about how common your disease is and how lethal it is, right? That's if no one would do a meta-analysis on the common cold because it's not lethal, it's not important, it's not deadly, right? But when COVID happened, there were a lot of researches on COVID, a lot of meta-analysis, right? So a topic is important for readers and journals if it's common or if it's very dangerous, right? So you show that very high mortality and so on, right? That's how you basically sell your topic in the introduction. Then you talk about what has already been done on that topic, right? And you move towards your specific topic, and then at the end you write down the rationale and the objectives. We're going to discuss this with an example. That's the easiest way. So the way I talk about this, the easiest way to write a discussion for a systematic review is in the first paragraph, talk about your population and your comparison, right? If you think back to the PICO's framework, talk about your disease basically, and you talk about the comparison, basically the treatment that has always been used. For example, in the radiation versus chemo example, we are going to talk about leukemia, how common leukemia is, how dangerous leukemia is, and so on, right? And then we're going to talk about chemotherapy, that chemotherapy has always been used for leukemia and so on. In the second paragraph, you will talk about the new intervention. So now you talk about radiation, how it works, what's the evidence behind using radiation, and so on. And then in the third paragraph, you write down your rationale and your objectives. In the rationale, you talk about any previous meta-analysis that have been done on this topic, right? And how is your paper going to add to that, right? If there's already a previous meta-analysis, how is your paper different? And the objective is simply your title written as a sentence. So if we look at this example, so this research was actually on, dialysis patients, and we were comparing two types of balloons because dialysis patients have this issue that their vessel closes after some time. So you need to keep that access open, right? So the introduction, it starts off talking about how common CKD is, how common dialysis is, right? Like it's one of the most common issues, right? It's a major cause of morbidity, mortality, and costs, right? And then I talk about how common it is for the dialysis access to close. That's basically my population, right? Patients with a dysfunctional dialysis access. Hopefully that makes sense. I know this example isn't very helpful for second, third year students, but this is my population, right? This is the disease I am working on. So that's the first paragraph. Then you talk about the new thing, right? So advancement in technology has caused this. This is my intervention. So now I'm talking about how my intervention works. In the simpler example, this would have been radiation, right? How does radiation work and so on. And then the last paragraph, this is the rationale, right? It's pretty small. Usually you write it longer, but the goal is you talk about any previously published meta-analysis and how is your paper different. So maybe in the other example, we have something better. Yeah, so in the other example, this is again the last paragraph. First paragraph is on the population and the comparison. Second paragraph is on the intervention. Third paragraph is rationale and objectives. Several previous meta-analyses have been done on this topic. However, new RCTs have been published in recent years. Furthermore, this specific thing has not been seen in previous papers, and we are looking at safety outcomes, the safety of this drug in more detail. So I just listed down things that my paper has that the previous meta-analysis did not have. That's another way to sell your paper, right? So everyone who reads it thinks it's really important. So that's what the rationale is. Talk about the previous meta-analysis and how your paper is different, better than them. The last sentence is objectives. I'm just going to edit this. So the objective is simply, we have undertaken a systematic review and meta-analysis to assess the efficacy and safety of this drug for this disease. For our radiation versus chemotherapy example, the objective would be, we have undertaken a systematic review and meta-analysis to compare the efficacy and safety of radiation with chemotherapy in acute leukemia, right? So simply your topic, that's your topic, radiation versus chemo for leukemia. That topic, just write it down as a sentence. That's the last sentence, that's the objective. After that, supplementary file isn't that important. The abstract, it's just a trailer for your research. So it provides a brief overview of your objective, method, results. So let's look at an example of the abstract. So in the abstract, again, there's subheadings. Background is basically a summary of the introduction. Methods is obviously a summary of the actual method section. Result is a summary of the result section. Conclusion is a summary of the discussion section. Keep it to 250 words. In the background, just 2 sentences. One sentence is just a quick recap. We are looking at this population XYZ. The second sentence is the objective again, right? This meta-analysis compares this with this for this disease, right? In the method section, just some of the details, like you use some databases, what was the date, what was the inclusion criteria, what was the analysis like, right? So just choose some details and briefly summarize them. In the result section, how many studies were included, and then just talk about your actual meta-analysis results, right? For example, for this outcome, the 4 values again, right? And then so on. Just talk, you don't talk about study characteristics and all that, just talk about how many studies were included, and then jump straight to the actual analysis results. Finally, in the conclusion, a quick recap of your results, that this outcome was significant and this was not significant, and then further research needs to be done to look at XYZ, right? So this is the abstract. So keep it to 150 to 250, keep it to 250 words. 150 is a little small. It's basically a hook, and I also called it the second most important section in a paper. So this is second most important because this is the one so it's basically based on how many people read which section, right? So the abstract is the one that most people look at, except for one, which is more important. So I just want you to guess what the most important section in a paper is. The one that gets read the most. The title, that's the most important thing, right? And it's easy to make. Just use your PICO's framework and make it in a very concise manner. So the example I was using, total body radiation versus chemotherapy in leukemia, right? And then a systematic review and meta-analysis, right? So intervention versus control in this population, a systematic review and meta-analysis. So that's the title. A couple of quick tips. Don't use too many words. Don't cite every article. This is an interesting story. I found that the structure of DNA was published in a 1-page article, the entire structure of DNA, right? So if you start talking about if your research is on heart attack, you don't need to say that this vessel closes and the anterior descending closes, and this is how atherosclerosis works, because you are writing this for other medical students and doctors. They know this, they have read this as well. You're not the only one who studied this. So just focus on how common the disease is and then jump straight to your topic, right? Don't exaggerate. For example, no previous study has assessed this outcome. Don't say this, because you can't be sure. Maybe there was a study. So you said, to our knowledge, no previous study has assessed this outcome, right? Use formal language, but don't use too complex or language that no one would understand, right? The analogy I gave is the way you look at news reporters. They just state everything, they leave the interpretation to you, right? There's like talk shows where they interpret everything, but the actual news report that comes every hour, they just state everything. That's the tone you want to use. Very formal, very serious, right? And then just use an outline. You have to connect each sentence, each paragraph with the next one, follow the flow, and that's it. So before we end this, I'm just gonna, so another thing I had to cover was Prospero, right? So I'm just gonna cover Prospero, and then after that, if there's any questions on, and I understand that this is the last webinar, so if there's any questions on any of the steps, because I promise I can do all of them, so we can discuss that too. So Prospero is basically a place you can register your topic, right? It's like a registry. If you have a ERB in your institution, I don't know where most of you people are from, but from RMU, where I'm from, where I was from, they have an ERB, right? That's where you go, you present your synopsis, you register your topic, right? I don't know if you guys have that. Maybe you can let me know if you have that as well. But you make a research proposal for any cross-sectional study or anything, and you present it, it gets approved, it gets saved in the database, right? So no one else does this paper. In the same way, the meta-analysis, you don't need to register with your institute. They have no say over what you do with your laptop, right? Because you aren't using data from your institute, you're using available data. But you can register in Prospero. Optional, but highly recommended. So this is Prospero. You make an account, you log in, close. So this is my Prospero. I have registered these 20 papers from 2024 onwards. So the way you register your project is create a new Prospero record, right? So you have a topic in mind, you want to register it. So it's going to say, I agree to the terms and service. And now it's just like a form, just fill in the form, right? Don't, we're not doing research on animals. So we're going to select humans. This is not part of our training course. So we're going to select this option, right? We are doing an intervention review, right? Intervention review basically means you're looking at treatments and stuff. That's the only thing Prospero has right now. Everything else is not even available. So it's not really a choice. Then you click on register your review. And now this is a form you need to fill in. So here you write down, this is the first category. Write down the title. I'm not going to go over all of this, but you basically write down your title here, right? And then you can save everything. Here you write down which domain is being studied. So it always gives you examples. So it's really easy to use, right? You don't have to get worried. For example, my disease. So my disease is acute lymphoblastic leukemia, right? Acute lymphoblastic leukemia. That's a type of blood cancer, right? This is my disease. I can save it, right? So this is my disease. What's the rationale? The rationale is the same as the introduction. So sometimes when I'm lazy, I do the registration after I have written the manuscript, because then I can just copy from the manuscript, right? I can just copy my rationale from the introduction, all of this, and paste it here, right? So the meaning of rationale is the same. So you write down your objectives, you write down which country you are from, you write down your inclusion criteria. I'm not going to go over all of this, but we have covered everything, right? You just need to fill in the form now. Any similar reviews? What's your timeline? So you can just mention everything. I'm starting today. I'm gonna end 21st August, right? Whatever. So you write down screening results here, and you write down data. So whatever you don't have, you can just say we haven't done this. It's fine. They'll still accept your proposal. They don't really care. They don't even read this. They just the computer, the AI just accepts everything, and you just fill it in, and then at the end you can save this, right? So I'll show you what a complete example looks like, because I can't fill this in because it's going to be really boring for you and it's going to take a long time. So this is a complete example. I want to see one of my versions. They keep changing. View record, right? So I pasted the title. This is my objective. This is the search I'm using, PubMed, Embase, Cochrane. This is my inclusion criteria. This is my disease, heart attack, myocardial infarction. This is the inclusion criteria for population. These are my interventions and comparisons. You talk about your outcome. So basically every step you have to write down here, right? And I just copy pasted from my manuscript. So talk about data extraction, talk about your analysis, who were the authors. You don't have to mention all of them. You have to mention a minimum of 2. The timeline, and then that's pretty much it. Country is Pakistan. So question by Irtiza is, when do you have to register? The right thing is to do this first. This technically should be the first thing that you do. But a lot of times when you start a project, you aren't sure what you're going to get, right? Maybe you need to change the inclusion criteria. You only included RCTs, but there were not enough trials, so you had to include cohort studies too. So you need this flexibility. Once you submit this and it gets published, you don't have that flexibility anymore. You are now bound to what you wrote down here. So even though technically you have to do this first, I do this near the end. When my manuscript is done, they take 2 weeks to review and approve it, and then as soon as they approve it, I submit it. So that's why I do it at the end, because I like to have that flexibility. But the honest answer is that you should do it first. Hopefully that makes sense. Okay, so that's Prospero. Now if there's any questions, feel free to ask them about manuscript writing or any step. We can cover that. If we do it first, how much do we have to be able to fill out, considering you mentioned we can write not done yet in sections? You have to fill in everything. It can't be submit. Some of the fields are mandatory, like data extraction. You have to mention this was all the variables that I extracted. In analysis, you have to mention these were my outcomes. So one thing you can do is you can always amend your records. For example, this is a published record, right? I can make revisions here, but it just doesn't look good because this is publicly available. So, people will see you kept revising your stuff, you kept changing it. So that's why I do it at the end. But you really need to, pretty much you need to write down everything. You need to tell them on the very first day. You need to know what mesh terms you are using, what databases you are using, what software you'll use for screening, what will be the variables you extracted, how will you do risk of bias, how will you do the analysis. So basically everything needs to be told. Should the Prisma flow diagram be included in the main manuscript or as a supplementary file? So supplementary file is basically, for some journals, they have a limit on how many figures or tables can be included, right? They have this limit. So that's why what I do is I try to include, like, a manuscript, a meta-analysis has so many figures. All the forest plots are separate figures, right? So if the journal says only include 3 or only include 4, some journals have no limit, so it varies. What I do is I include, I'm just explaining what a supplementary file is before I get to your question. So what I do is I include the minimum number in the actual manuscript, and then the remaining figures and tables are added to a supplementary file. That's like a Word document that you upload directly. It's like all the extra stuff, right? All the supplementary stuff. Upload it directly, and the journal doesn't have to print it. So it doesn't, they don't have to spend money on it. They just add it as a link and anyone can download and view it. So that's why a supplementary file exists, to meet all the journal criteria, and journals sometimes have extra charges for extra figures. So with the supplementary file, you can avoid all of that. How do you divide your figures and tables between supplementary file and main manuscript? The very important ones go to the main manuscript, the ones without which the paper is incomplete. And then the extra stuff that if someone wants to see, they can, goes to the supplementary file. But I would recommend is, the first figure is the Prisma flowchart. Okay, hopefully you guys understand that, figure 1, figure 2, they're written like this because this is the order they appear in, right? Figure 1, Prisma flowchart is figure 1 only because this was the very first figure in the manuscript, in the way this is written, right? This is the first figure. So that's what's called figure 1. So the Prisma flowchart goes into the actual manuscript, right? Prisma flowchart will go to the actual main manuscript because there's only one Prisma flowchart. You're summarizing 2 of your most important steps in the Prisma flowchart. So always include the Prisma flowchart in your main manuscript. The study characteristic table also goes to the main manuscript, right? Because is summarizing all the studies in one table. That goes into the main manuscript. Then the forest plot for the primary outcome or the most important outcome. In this case, this was my primary outcome. So this went into the main manuscript, the forest plot for the main or the most important outcome. All the other forest plots for the secondary outcomes will go into the supplementary file. If you did risk of bias assessment, that will also go into the supplementary file. So Prisma flowchart, forest plot of primary outcome, and the study of characteristics table goes into the Main manuscript and everything else can go into the supplementary file. Hopefully that makes sense. I think I also answered your second question. Okay, Mehreen is asking, can you guide about bias management? I don't understand. Maybe if you could rephrase that question. Okay. I don't think there's any more questions, so I guess that's it.
Guys, if you have any more questions, you can ask them right now. I think we're done with questions now.
Yeah. Okay.
I'd really like to thank you so much. आपने इतना अच्छा एक session दिया. You did an amazing. You gave amazing explanations and you made such a dry topic extremely exciting. So I'd really like to appreciate you for this.
Yeah. No worries. Okay. Let me
Also, to
just.
all the participants, thank you so much for joining us throughout all these sessions and for staying with us till the end. The form will be shared soon. Post-evaluation form will be shared in the chat, so please stick with us and fill that. The form will close in 10 minutes.
Okay. Thank you for having me. Bye.
Thank you so much for joining us and for giving such an amazing session again.
Thank you.
The post-evaluation form is in the chat right now, so please fill it in the next 10 minutes.